Okay, so this is the second mini lecture from chapter 3, and it's going to pick up kind of after protein synthesis and talk about the different ways cells produce different cells. And so last time we talked about the nucleus and that the DNA of the cell is in the nucleus and it controls cell activities primarily by being made into proteins. And so the DNA code is the same code in every cell. And what happens is the parts of the DNA molecule are read as a code and turned into amino acids. And so three nucleotides, a sequence is a single codon that codes for one amino acid. And then the ribosome links the amino acids together based on an RNA messenger molecule. I also mentioned the replication of DNA, that because the nucleotide bases pair, if you know one strand of the DNA molecule, you can easily reconstruct the correct sequence of the other strand. And so the old strand serves as a template for making the new strand. And so DNA molecules are easy to copy, and they're copied exactly. And this explains why it's so easy for cells to divide and reproduce. And so the main thing in this lecture is the cell cycle. The idea that cells go through a particular pattern each kind of lifespan of the cell, where they grow, copy their DNA, prepare for mitosis, and then they divide the DNA after it's been copied and split into new cells. So we refer to the normal activities of a cell as interphase, and that's divided up into two periods: growth 1, synthesis S, and then G2 is growth 2. And the cell looks normal. It has a normal nucleus then, but it prepares for copying the DNA. And then after the DNA has been copied, it gets ready to divide it during mitosis. And mitosis divides the DNA into two nucleuses. So this is what happens in cell division. Here there's a long explanation for that, but the main thing is the appearance of the cells and what's going on. So in interphase, the cell has a normal appearing nucleus. It grows. It takes care of all its normal functions. So if it's a skin cell, it makes skin proteins. If it's a muscle cell, it makes muscle proteins, and it divides or it contracts and things like that. During interphase, though, at some point, if the cell decides I'm going to divide, it copies its DNA. It still appears to have a normal nucleus, but now it will have twice as much DNA in it. Okay. When it's ready to divide, the DNA gets tightly wound up into chromosomes. Each chromosome is made up of two chromatids. It is a copied DNA molecule that's linked together in the centromere. These first appear in prophase, and in prophase, the nuclear membrane disappears. The centrioles start to split apart, and protein spindle fibers form between them. In metaphase, the spindle fibers become attached to the chromosomes and line them up in the center of the cell. And so here you see the diagram on the left, and on the right you see the light pink spindle fibers, the dark chromosomes in the center, and the centrioles on either end. Okay, to make sure that each daughter cell gets the right amount of DNA, then each duplicated chromosome splits. Okay, and identical chromosomes migrate in the opposite directions. This happens during anaphase, and it's the spindle fibers shorten to pull these things apart. When they get almost all the way apart, we refer to that as telophase. And at telophase, generally the cytoplasm is starting to separate into two distinct cells. We call this a cleavage furrow, and we can see two nuclei starting to reform. Okay, and so the end of mitosis, we wind up with two cells that appear pretty much identical and have identical DNA. And so this mitosis allows us to produce extra cells. This is what happens during growth. Okay, but not all cells are growing all the time. Different cells will remain often in the G1 phase for much of their lifespan, sometimes all of it. There are checkpoints here that kind of determine the cell's fate. These checkpoints happen when certain compounds accumulate, and if they don't accumulate, the cell won't pass the checkpoint. Some of our cells will never pass these checkpoints, so they'll remain in interphase their entire life. Okay, but if they get past the G0 checkpoint, or the G1 checkpoint, excuse me, they'll copy their DNA. Okay, and then they'll go wind up through the G2 checkpoint, okay, where there'll be some checks to make sure that the cell is the appropriate size, that DNA replication has occurred correctly, and then mitosis will begin. At another checkpoint, there will be proteins checking to make sure the chromosomes have lined up correctly. At these checkpoints, if something's wrong, the cell cannot proceed. The cell will be stopped, okay, and often the cell is then eliminated if something's wrong. Cancer happens when damaged cells pass the checkpoint, okay, and these damaged cells have the checkpoint proteins aren't working, and so the cells are allowed to reproduce when they shouldn't. This is what causes cancer, okay, and so it's a major disruption in the normal mechanisms that control cell division. Okay, and so this leads to uncontrolled cell division, abnormal growth, tumors, cells that aren't doing their job. And this is usually caused by chemical damage to the DNA that leads to damaged checkpoint proteins. Cancer is a major problem. It causes 25% of all deaths in the United States. Fortunately, this mostly happens to older adults. Okay, and the damage is usually caused by exposure to chemicals or radiation. Sometimes these chemicals are in the diet. Sometimes they're exposed to them on the job, like asbestos. Sometimes things like cigarette smoke or high temperature chemicals from grilling that cause that. But it can also be radiation from sunlight or viruses. So we learned a lot about the cell cycle. Primarily, one of the main reasons is to figure out how to stop cancers. Okay, there are other applications as well. Those applications include trying to get cells to divide when we need them to divide. So some cells don't want to divide after like spinal cord injuries. This is Christopher Reeve. He had a spinal cord injury after a severe fall while horseback riding. If we could get cells to divide then when we normally can't get them to divide, we could cure many of these diseases and injuries, and we could heal transplant patients and stop degenerative diseases. I should also here mention that this cell division is also a time when cells develop specialized structures and functions. And this often results from the selective activation or inactivation of certain parts of the DNA chromosomes. Certain genes are turned on and certain genes are turned off. This controls which cells are active, and this primarily happens at the promoter region. So before a gene is going to be made into a protein, there's a region called a promoter. And the enzyme that's going to copy that gene into a piece of RNA is called RNA polymerase, and it recognizes the promoter. Okay, these promoters can be permanently turned on or permanently turned off. They kind of act like on and off switches. And so this is why some cells make certain proteins and not others. And so differentiation is really turning genes on and off.